So welcome everyone to this week's edition of the BPPB. So please remember to keep your audio muted and please enter your questions in the chat. We will ask them in your behalf. If there's an urgent clarifying question that you want us to interrupt the speaker for, then please try to indicate that if you can. But we really invite you to stay on for an informal discussion after the talk, unmute yourself, and then have a longer, more intimate discussion. It's a great pleasure to have Dimitrios Vavylonis, who's going to talk about cell patterning for division and growth. So Dimitrios, take it away.
Thank you, Ashok, for the introduction. It's a pleasure and honor to be part of this series. So I'll talk to you about some work we've been working over the years and to understand the molecular mechanisms of cell division and cell growth. So I'm at Lehigh University, and for those of you who are in the East Coast this morning, this is not a map of the earthquake that happened just 40 minutes ago and was felt in the area. We're about 30 miles west of it. I hope everybody is okay. I didn't hear of any damages. It was a 4.8 earthquake this morning, so you may remember this day. So my interest is understanding how this cells growth and divide. So here, this is a movie showing a fission yeast cell, which is a tubular cell, and this cell grows from the two tips and divides every few hours in the middle. It does so by establishing a zone of, it's called polarization. This protein called CDC42 assembles at the tip of the cell. And then upon division, you can see how this myosin protein assembles into a ring in the middle of the cell. So what is shown here with fluorescent markers is the spindle pole body that divides into two during cell division. And you can see the ring forming in the middle of the cell. It's made out of actin filaments and myosin motor proteins. So this polar growth and division is perhaps the largest scale pattern we observe in these cells. It's one of the simplest eukaryotic cells, so we can use it to understand the molecular basis of life. We have a case here where we have micrometer scale patterns that are made out of nanometer scale objects. So there should be physical aspects related to these processes, but we don't know what's the right level of abstraction and description to describe them. And this is important when we think about engineering or applications to reconstitute living-like systems, and more generally to understand how the genotype of cells is translated into phenotype. Okay, so as a question of pattern formation, I can connect this behavior maybe to two examples of physical phenomena that involve pattern formation. So here to the left is a pattern formed by spinodal decomposition. So this is a system that is approaching an equilibrium, and on the way we have this pattern that can be long-lived. So this would be a pattern towards equilibrium. Another mechanism that describes a pattern away from equilibrium that we've encountered many times in the series is the formation of patterns through reaction and diffusion. So we have an activator and inhibitor. They interact in a nonlinear way that amplifies local concentration fluctuations of the activator, and then this can create a local accumulation of an active form. And this pattern is driven by the consumption of chemical energy to create a pattern away from equilibrium. Okay, so how? Okay, let's look into the question of how does a ring form in the middle of the cell. So I have to tell you a few things about actin. So actin is a protein that exists in monomers and filaments. Its polymerization is triggered by, in purified conditions, by the addition of salt. So we have a step which involves a nucleation step. So three monomers come together to make the nucleus of a filament. And then these filaments can grow by addition of monomers primarily to the barbed end of the cell, to the barbed end of the actin. So if you look at experiments measuring and models measuring the fraction of polymerized actin as a function of time, we see this exponential approach to the equilibrium of fully polymerized actin approximately, or in lower concentrations, we have this lag phase at the beginning. So, I mean, the topic of ring formation in vitro has been fascinating to researchers in the cytoskeleton area for a long time. And we know that actin filaments, being flexible, can fold up and make rings on their own in conditions where we have a cross-linking protein that links the filament together, or in a poor solvent condition, as shown here. In confinement, in micrometer size confinement, similar to the yeast case, the polymerization of many actin filaments in a confined droplet or vesicle can bundle the actin filaments into a ring, so provided the confinement size is very small. And actually, there's a talk next week about this topic by Alan Liu. Okay, so could the ring formation in yeast then be as simple as the approach to equilibrium of a bundling actin filament system? So it turns out it's not as simple. So, and I'll just make a note that kinetic effects are very important in these systems, and I'll demonstrate this with a simulation, a Brownian dynamic simulation we made recently with collaborators at Flatiron Institute, showing a condition similar to the experiment I just showed, where we have polymerization of actin filaments in a confining environment in the presence of an this alpha-actinin cross-linking protein. It's a passive cross-linker. And you can see how we get this bundling of these filaments together, and then the slowly merging and coalescence of bundles that give rise to a ring in times very comparable to the experimental times. However, if all the actins start pre-polymerized in this simulation, we see something very different. We see the bundling of these filaments into a structure that gets trapped into a metastable state as it slowly coarsens, that turns into a structure that's different from a ring. So kinetics are important when we deal with many filament situation, as in the case of the contractile ring. So the actin system is regulated in many ways, and partly we believe is to allow the system to undergo flexible transformations. So here I will introduce you just two of the among many regulators. One of them is this formin protein that binds to the barbed end of the actin filament. This protein is typical. It can be found in cells attached to the membrane, and it can add monomers to the actin by recruiting them to the end through a flexible domain. And in this way, it can both enable the polymerization of actin away from certain parts of the cell, like the membrane, and also regulate its elongation. Actin disassembly is also highly tuned in cells. So in these experiments, these workers had actin filaments on a slide, and then they flowed in this severing protein called profilin that binds to the sides of actin filaments and breaks up the actin filaments into pieces. So here in this experiment, these filaments are carried away by the flow away from the field of view. So we have this situation of constant assembly and disassembly of actin. And furthermore, this process is guided. So here I'll show you some experiments. So, by the way, I should say here that my research group is focused on mathematical modeling and computational modeling. So all the experiments I will show were developed in collaboration with experimental colleagues with whom we have looked into these topics and tried to approach them from a quantitative perspective. So here, this is a yeast cell imaged by confocal microscopy. And what you see is that these myosin proteins assembled into this structure called nodes that form in the middle of the cell. These nodes come together into a ring when, during the cell cycle, the polymerization of actin is initiated in the middle of the cell. So you can see here in this bottom experiment where the actin is labeled in green. When the spindle pole body in the middle of the cell splits into two, a puff of actin is created in the middle of the cell, and then this network of actin brings together the nodes into a ring. So we have now some understanding of the molecular composition of these nodes that are attached to the membrane by this aniline protein MID1. There's another protein called CDC15, and then this brings this forming CDC12 and myosin together to create this kind of actin generation and post-producing machinery. So we've worked to understand the mechanism of this process. This is work with Tamara Bidon and Nicola Orkic, where we created a Brownian dynamic simulation of nodes, here simulated as point particles, that have this ability to create actin by polymerization, pull on the actin by motor forces, and then the actin also can link to other actins through cross-linking by proteins such as alpha-actinin that these cells have. So we call this model stress capture, pull, and release because these filaments grow, they get captured and pulled, and then through cophilin-mediated severing, they turn over and renew themselves. So this is the elements of this Brownian dynamic simulations, where each filament is represented as a series of bead connected with springs. They have a certain stiffness and bending rigidity. And then when these filaments are near each other, they can attract each other through these passive cross-linkers that we describe here approximately as an effective radius of attraction and depth of this interaction. We can see how now if we start this simulation from by placing nodes, these red dots, in the circumference of a cylindrical domain, over time, we see how this polymerization of actin from these nodes, the bundling together by this implicit cross-linking assumption we have, and the pulling of the actin by the motor proteins that are in these nodes can lead to this actin bundle that self-organizes into a ring-like structure. So we've developed this model by looking in detail at how these nodes move in the cell and analyze the pattern of this type of condensation process. I mean, and this is a fascinating aspect of this model system, is that experimentals have ways to modify the composition of molecular composition of these cells genetically. So we can test some predictions that we can derive from such an approach. So the successful ring formation in this model depends on this cross-linking parameters being in the right range. So this is the behavior of the system as a function of the cross-linking interaction strength and depth. So if these parameters are very weak, then the bundling process is very weak. So in the simulation, we get aggregates of clumps of nodes. On the other hand, if the cross-linking is very strong, then the bundling process takes place too quickly. So we get this frozen-in double rings. And this is exactly what is observed in cells, where in the left side here, the alpha-actin protein is expressed at high levels by changing the promoter of this alpha-actin protein. And then even though the nodes start uniformly, they collapse into this kind of double ring structure through which the cell has a way to deal with it later on by constricting part of it. On the other hand, if the alpha-actinin is deleted, removed from the cell, and then we have this other cross-linking protein called fibrin is expressed at a lower level, then these nodes coalesce into clumps. So they fail to make a nice ring at the beginning. Though later on they have ways to repair that process. So I hope I showed you in summary how this ring assembly in this yeast cells with understanding it as being guided is a process of bundling of actin, and turnover of actin, but it's guided by these nodes that form in the middle of the cell. Okay, but then how do the nodes form in the middle of the cell? So this is a basic question related to this this medial division pattern. Okay, so we know that the nodes structures actually exist through the whole cell cycle. So this experiment to the left shows one of many experiments that have looked into the behavior of nodes through division. They help to serve not as a scaffolding for the ring, but also as an antenna for the cell to sense their size and regulate their cell cycle. So it's a very fundamental aspect of these living creatures. And one of the early proteins that come into these nodes is this protein called CDR2. It's a kinase. And you can see here, after division, it creates these nodes in the middle of the cell. One mechanism that's been established about how this takes place is through inhibition by another protein called POM1. So POM1 is another protein that assembles to the tips through this tip formation mechanism I will talk about later, parts of it. Okay. Okay, so we've been looking at this mechanism, and our collaborators in the Mosley group at Dartmouth came to us and to tell us about their discovery that this CDR2 is actually going in and out of the nucleus. So there's nuclear shuttling. So this is an image of the cross-section through the middle of the cell. And there's some intensity of CDR2 in the nucleus, very small, but it becomes more clear if this protein called R6, which is a stabilizing protein of CDR2 in nodes, is deleted. And you can see there's more of it. You can further increase the concentration of CDR2 in the nucleus by treating the cells with this nuclear export inhibition drug called leptomycin B. And it turns out this also depends on this other node protein called MID1. This is an aniline-like protein that's also found in regulating cytokinesis in animal cells. And this nuclear shuttling seems to play a role in positioning these nodes, because in the cells that lack this R6 protein, when the nucleus happens to be close to the one side of the cell, you see these nodes form near the side where the nucleus is close to the membrane. Okay, so this gave us some clues about the possible mechanisms. So we tried to create maybe the simplest model I can think of to create this node pattern that involves inhibition by POM1 at the tips. And we use this a particle-based simulation based on Smolden software to simulate the system, assuming now that the CDR2 can be in two states, active and inactive. So this is part of the still not understood cell signaling system in the cell that regulates the activity of CDR2. So these active proteins of CDR2 can bind the membrane. So CDR2 has a membrane binding part, and we assume they can stick together to make an aggregate through nucleation and growth. So they can go in and out of the nucleus. In the nucleus, they can be activated, and they can be deactivated by POM1. So we can get some reasonable numbers for this diffusion and numbers of the cells from prior measurements. So what we find is that this is a simulation of this system. You can see this CDR2 accumulating as nodes, these are the green dots in the simulation on the membrane. So we assume these are static as well. And in the bottom part, this shows the equilibrated simulation after 50 minutes. So this a pattern is very similar to the cellular pattern, provided we tune the two main unknown parameters here, the growth constant of the nodes and then the nucleation rate constant. So by tuning these parameters, we can get the approximately right number and the size of the nodes. Okay, so maybe this is an aggregation process that's driven by some signaling system in the cell. But we want to test this. Okay, if the nucleus is really an important pattern formation, important player in this pattern formation process. So first of all, we want to see if this model can explain the observation of higher number of nodes when the nucleus is displaced to the one side of the cell. And we do observe that in, so this is a simulation of wild type cells. And then in the cells where the nodes are more dynamic because they lack this protein called ARF6. And you can see here in this simulation, I hope, that initially the nodes are more uniformly distributed in intensity. And then in 10 minutes, their intensity increases when they're closer to one side of the cell. And this happens both for wild type and ARF6 delta cells, but faster in the ARF6 deleted cells, presumably because the nodes are now responding faster to the fluctuations of the nucleus within the cell that occur over the minute time scale. Okay, so now, though a prediction of this mechanism here of coupled POM1 and nuclear activation would be the displacement of the nucleus to the outside of the cell. So we would predict that if the nucleus is moved to the one side, over time, the nodes move by rearrangement of the numbers in each node, and also there's a slower process of disappearance and formation of new nodes. So Hanno Palko in the Mosley group did these experiments by depolymerizing the microtubules that help center the nucleus in these cells, and also move the nucleus by centrifugation. And I hope you can see that when in wild-type cells, the nodes form around the nucleus. So this is a marker for both this MID1 aniline protein and CDR2. When the nucleus is displaced, a big part of the nodes rearrange within time scales similar to what we would predict in the model in about 15 minutes. Okay, so there's some ideas now. We have some kind of way to start thinking about this node pattern, but this involves, I mentioned this process called activation, and how can this take place? You know, what is this type of activation? So we don't know the details of this process yet, but we have some idea now looking at another protein called CDC15. It's a protein that is essential for the cells. They die without it. And it joins the nodes and is important in cytokinesis after CDR2. It's the protein that is a main contributor to bringing the formin to the nodes that creates the actin filaments for cell division. This protein has this, it's called the F-bar domain. It's this banana shape. It's formed by a dimer of these proteins. Interestingly, as many of these node proteins, the globular part is only a small fraction of it. Most of it is a disordered region. It's a flexible, floppy chain, as far as we can tell, as many of these node proteins tend to have, including CDR2. So there's work by many people has shown how, in particular the Gould group, has shown how this CDC15 can exist in active and inactive form through a process called phosphorylation of the disordered region. So phosphorylation of the additional phosphate group, a negative charge group, to the disordered region. Okay, so charges seems to be important in this activation mechanism of one of these node components. So, okay, if you look at the charge distribution of this F-bar protein, it's mostly negative in the middle, and then the ends are have a positive charge. This is where the membrane binding residues are located, the amino acids. And also the residues that help this F-bar oligomerize and make a scaffolding on artificial membranes, and presumably within the nodes, though we don't know the precise architecture of CDC15 in the nodes. So the Gould group and Rahul Bhattacharjee in particular did a long study to map out all the locations where this disordered region gets phosphorylated, or nearly all. And by a combination of mass spec methods. And you can see that what they found is that many proteins, including POM1, the protein that regulates CDR2, but also a few others like PCKN1 and SHAKE1 and KIN1, help add these negative charges to this region. And there's really a lot of them, like 35. If you mutate these residues so that to become alanine so that they cannot be phosphorylated, then the behavior of the CDC15 in nodes seems to be determined by having sufficient number of phosphorylation sites. Okay, so we figured, okay, if it's something as largely driven by how many of them we have, maybe you can simulate that. And for that, we used recently developed models for disordered proteins. These are coarse-grained models. This can only be approached at this level by some kind of coarse-graining procedure. And in this molecular dynamics model I will show you, we replaced every residue by one bead and used this the HPS model developed by Dignan et al., who calibrated the pairwise energies between amino acids to capture the radius of gyration of these disordered proteins for and then tune this to match the experiment. So this model has the effects of phosphorylation have been included. So by Perdicari et al. So okay, so this is now a simulation that Aaron Hall in my group did, where he took the non-crystal structure of the F-bar dimer of CDC15, and then added this disordered region as flexible chain to the two ends. Then there's further another globular domain called SH3 is at the other end of this region that is important to connect the CDC15 to other proteins in the nodes. So this is the simulation of this model. When the disordered region is fully phosphorylated, we have all these 35 amino acids with a negative charge on them. And you can see what happens in the simulation is that these floppy regions tend to hover around the positively charged end of this F-bar region. And while they can reach out to find each other, they are not so interested. they have similar charges. When now we remove all these charges, we can see a dramatic effect in the simulation where they will go from a segregated disordered region to the ends of the F bar to a collapsed configuration of this disordered regions towards the middle. So the loss of the charge by phosphorylation makes this disordered regions less interested in the positive charge near the end of the F bar. Okay, that's part of the elements in the simulation, and more interested towards each other. So they can also extend more, they become more extended in size. So they seem to make something that's more of a phase separate, or the elements of the nucleus of phase separated objects when in a many protein situation. And this would allow the F-bar proteins to stick with each other and also stick to the membrane. Okay, so this is a simulation showing how this adding negative charges here, these are the yellow dots, expands the disordered region. The size of the disordered region is a signature of their tendency to phase separate. The more compact they are, the more likely they are to aggregate with themselves and each other. So then Rahul took this suggestion and then explored the phase separation properties of CDC15. And indeed, it purified CDC15 when it's fully dephosphorylated. It's purified in the presence of proteases. Then they can form at sufficiently low salt concentrations. They make aggregate into droplets, and these droplets fuse, exhibiting the liquid-like nature, while the phosphorylated form does not. And this is elements of this phase separation properties is also seen in cells where expressing a protein that cannot be phosphorylated forms aggregates on the membrane of the cell, and they seem to fuse in live cell images. Okay. So we have, it seems that the node assembly then in itself seems to have physics in itself. There's some aspects of this, for sure not all aspects of it, seem to be driven by the signaling system in the cell, triggering some kind of the tendency of proteins to go in and out of liquid condensates that may help trigger some kind of higher order structure formation. That's needed for for this stable machinery to withstand forces in the cell by actomyosin. Okay, if I can pause here, I'm going to see if there's any questions. I can now move to the next part about the mechanism of polarized growth.
So there's one question, Dimitrios, about your search and capture and pull model.
Yes.
So Amrita wants to know what physiological parameter within the cell corresponds to the spring constant K between the cytokinetic nodes, and how is this parameter modulated by the cell? And actually, I wanted to add a follow-up to that, which is that do the polymer characteristics even matter for the model?
So here, how do I pick the I'm sorry, which parameters? The polymerization speed is important. That's right.
So, so Amrita is asking whether what physiological parameter within the cell would correspond to the spring constant K between the cytokinetic nodes, and how is this parameter modulated by the cell? Okay. Yeah, so this is the spring constant here. So it should depend on how many cross-linking proteins we have, and what type of cross-linking proteins there are. So this is
So there's different types of cross-linkers that can be this alpha-actinin cross-linking here is a long and flexible linker, while the fibrin is a tight and stiffer cross-linker. Yeah, we don't have at the model, at least in this cells, a good understanding of how the physical aspects of these cross-linkers play a role. And in the model, they are just approximated as with these effective parameters that represent the binding and unbinding of many of them over time.
Okay, thank you. And does the spring
Something the
constant
next step
matter?
of this model that would require like more detailed simulation? Yes.
And does the spring constant matter for the results? I mean, if you presumably
yes, it matters.
yes.
Yes, this is what is shown here. Yeah, if it's very high, for example, they can get frozen into metastable states like stars. They can get frozen in very quickly if that's irreversible. Or nearly so. Okay.
Okay, so Amrita has a follow-up, but maybe we could take that after the remainder of your
Okay, all right. Thank you. All right, so now, okay, how the cells sense the tip, okay, establish a tip. So here, main player, one of the main players is this protein called CDC42. So this is a small GTPase. It can bind the membrane, and it exists in two forms, an inactive and active form. The active form can be inactivated by this inhibitor protein called GAPs, GTPase activating proteins, and there's 3 of them in these cells, and can be activated by proteins called GEFs, and there's 2 of them in the cells, SED1 and GEF1. So there's this cycle in the cell that activates and inactivates them. There's a positive feedback and then some kind of negative inhibition. In regions where this CDC42 is active, which is near the tip of the cell, this is where the cell guides the secretion by exocytosis and promotes the buildup of a cell wall that encloses the cells and the growth of the tip. This protein is also oscillates from tip to tip in mechanisms that are not completely understood. And yeah, it's one of the perhaps the most important protein in cell shape, because mutations of its regulators have a profound influence on the shapes of these cells, as you can see at the bottom. Okay, so this is this process is thought to be an example of a Turing type pattern formation mechanism, and this is more general, relevant for small GTPases. So this is an example of patterning during morphogenesis in oocytes. In yeast cells, more studies have focused, or many studies have focused on budding yeast, where the CDC42 makes one patch that's helpful for the creation of a new bud, the formation of the new bud. And this is thought, the model by Goratsev and Bokilko describes this as a winner-take-all mechanism. The positive feedback takes on, and then all the activators of CDC42 coalesce into a single spot. Okay, however, I mean, we know from fusion yeast that these inhibitors are also important, and they have a pattern on their own. So while the active CDC42, this is visualized here by this crib marker, is at the tip of the cells, it can be at both tips or one tip. Its inhibitors, 2 of them, the RGA6 and RGA4, accumulate away from that. So this is RGA4 on the sides, RGA6 on the sides, and RGA4 also on the sides, while RGA3 is at the tips. Okay, so there's some pattern of inhibitors that's important for the cells. If experimentals remove all 3 of them, the cells now become completely round, essentially. And you can see the activation of CDC42 pretty much all around. Okay, so we've been trying to understand with our collaborators the mechanisms of this pattern. And Deneta Giganova here in the Sophie Martin group made a crucial discovery when working in this optogenetic system, where by CIBN and CRY2 proteins that can be made to stick together and oligomerize on the membrane by blue light. So she had made this system in yeast cells, and in the dark, the CRY2 is in the cytoplasm while CIBN is on the membrane. And indeed, when light is shone onto the cells, you can see the CRY2 assemble into some kind of oligomers on the membrane, on the plasma membrane of the cells. But then she observed that when over time, this, and you can see maybe here in the image to the left, to the right, this CRY2 is slowly displaced away from the tips. So the tips get cleared from this protein. This is where the active CDC42 is located, as measured here by CRIB. And this displacement of this protein from the tips depends on exocytosis, the delivery of vesicles to the tips of the cell. So they asked us, is it possible that the membrane delivered here to the tips, can it somehow move proteins away from that site, membrane proteins? So we looked into this topic with David Rutkowski in our group, who developed this particle-based model. So representing the tip of a yeast cell here simply as a sphere. Okay. And we can assume we have particles that can bind and unbind with a rate constant K on and off. And if the cell delivers membrane near it, then this excess lipids would flow away from the site of exocytosis. So this is a top view of around the site of exocytosis. So we know the cells have their membrane seems very flat, so it must be under tension, so it will stretch out, and presumably carry the membrane proteins, the peripheral membrane proteins, with this lipid flow away from that site. And we also know that these cells deliver vesicles in a narrower area compared to the region where exocytosis takes place. Compared to endocytosis takes place. So endocytosis happens on a wider area. So there's actually a cycle of membrane, because cells deliver more membrane than what's needed for growth, and then they recycle it away from the tip. Okay, so by measuring these rates in experiments by the Martin group and Benetta, as I mentioned before, we can see that, yeah, indeed, we have flows with velocities that can displace peripheral membrane proteins, and this is the simulation here, when these proteins are bound to the membrane for a long enough time, and also they diffuse slowly enough. Okay. Because if they dissociate and bind in the cytoplasm, they probably diffuse much faster, but on the membrane, they could be slowly diffusing. So they created this fascinating experiment where now, based on this knowledge, they could transform the shape of the cells by light. So this is an experiment where working in this mutant that lacks all the gaps, so they're round, but has this gap protein that can be recruited to the membrane by light. It's called optogap. So when the cells are grown in the dark, the optogap is in the middle of the cell, and then the cells remain round. When they are grown into light, they are tubular in shape, because it accumulates on the membrane, gets displaced by the flow, and creates this deep growth pattern of fission yeast cells. In a time-lapse experiment where the cells are moved from dark to light, you can see how initially the optogap is, after exposure to light, binds the membrane, and then a site gets cleared up from this protein, and then a tip grows out of that site. Okay, so there was this discovery that then membrane flows here contribute to cell patterning. So there's something more than simple reaction diffusion by Turing type mechanism. There's the physical flows by lipids that are created by this pattern contribute to it. So how exactly does this happen is the next question then. So with David, we used models people have developed in the context of budding yeast to address this mechanism in the context of now the presence of this membrane flows. So this is a particle-based model that implements CDC42 in active and inactive forms, yellow and blue. In fission yeast, the inactive form of CDC42 is mostly bound to the membrane, but can also unbind. There's a positive feedback loop involving the activators of CDC42, the GEFs. And so this model here with just that, that's described, is known to create patches on the membrane. This simulation by Pablo. What we added to this model is the presence of a slowly diffusing gap and the flow, as I described, by this displacement of proteins around the sites of excytosis and endocytosis. So here we assume that whenever we have active CDC42 on the membrane, around that we will have enhanced exocytosis and over a broader area endocytosis. Okay. So now, okay, the association and dissociation rates are very important in this process, whether they will be influenced or not by flow. So we measured the parameters for CDC42 by FRAP experiments, fluorescence recovery of photobleaching. And this CDC42 binding and unbinding and diffusion seem to be fast enough to be away from the region where in this dissociation rate and diffusion coefficient map, you'd get this influenced by the flow. While the gap proteins are likely to be in that region. Okay, so now we can put this mechanism together into a model and see if we can create this Turing pattern combined with flow patch formation. And you can see how now an active patch of CDC42 forms. This is the region where we assume there's more local secretion, and that displaces the gap, which are the black particles here, away from that site. This is a projection of this sphere simulation in 2D. Okay, so now this is the things one can do with Pombe is kind of amazing, because what Sophie Martin's group did is to now change the mobility of CDC42 itself. Right? As I just mentioned, the wild type CDC42 is binds and unbinds to the membrane
Dimitrios,
fast enough. Yeah.
maybe 5 minutes more.
I'm sorry?
Maybe 5 minutes more.
Okay, yeah, I'm almost done. Yeah. It's in a region where it's not influenced by flow. Now, this CDC42 is bound to the membrane by a terminal region that gets a post-translational modification that is a membrane insertion that is just to the membrane. This can be replaced by another small peptide that binds to the membrane. It's called 1-RITC. And then the affinity of CDC42 for the membrane, as well as the diffusion coefficient, can be modified by adding more of that peptide, like 2 copies or 3 copies. And you can see how in FRAP experiments with diploid cells that have both wild type and this mutant CDC42, they could bring the diffusion of CDC42 into the range where the cells now must be displaced by flow. So what happens here is that these cells, and then now if you only have that copy, then the 1 Ritchie cells and 2 Ritchie cells lose their polarization and become rounder. And that's because the flow is the thing is displacing it. And this is what the model predicts, that with increased reduction in mobility, the paths will get weaker and broader. Eventually, it is lost. So this cell, the cells that have 3 Ritchie cannot live. We think this is because they have such a low CDC42 level that is beyond this survival threshold. So we would predict that if we maybe the cells care more about having enough CDC42 than that have a path. So by deleting the gap, we would expect, according to this model, would get more activation but less a wider path. And indeed, this is what is found when in a background where the cells have no gap, that normally will be normally dead. The cells that have the 3 Ritchie, the very slow CDC42, they become viable, in a background where they have no gap, but they're round. Okay. So this is, I guide you through various topics that I use the term pattern formation here in cells to talk about organization of a medial ring, and then how the nodes play a role, how these phosphorylations may be driving these nodes, and then also about the how membrane flows coupled to reaction diffusion systems in these cells and presumably in other cells to pattern them. Okay, thank you for your attention. Here's acknowledging of many collaborators and my current group. Thank you.
Thank you so much, Dimitrios. That was a fascinating talk. I just loved the way you began with, the early work on the cytokinetic ring, and then built it up part by part till you get this complex picture of this entire thing. So, folks, questions. There's maybe we can begin with Amrita's follow-up question. So what she asked was, and Amrita, feel free to unmute and ask if you prefer to do that. Okay, so I'm going to just ask it on her behalf. So what she asked was, does it mean that in the model, the spring constant only appears when the nodes come close enough to be linked by cross-linking proteins? Is it Brownian motion?
That's right. Yeah, it's a combination of Brownian motion and also the forces by polymerization can displace filaments around. That's right. Yeah, this actin filaments, as they grow, they make cables, and you can see them. They fluctuate in the cells. Actually, yeah, probably these movements are largely active movements, I would